Okay, you guys, this is the last video for the 6.2 notes. In this video, we're going to look at topic 4 and focus in on how genes are regulated in eukaryotes. So in the last topic, we looked at how gene regulation takes place in prokaryotes and bacteria cells, and how they control which genes are on and off and when those genes are being used or not being used. In this video, we're going to look at eukaryotic organisms, so like plants and animals and you guys, and how your guys' DNA and the genes in your DNA is regulated, and how in eukaryotic cells we can control when and if certain genes are being used. So in eukaryotic cells, this is important for a couple different reasons for gene regulation. Well, in prokaryotic cells, a lot of times their genes were being regulated depending on environmental stimuli, like depending on what's going on in their environment. Bacteria cells and prokaryotic cells, they can respond by turning on certain genes or turning off certain genes. We saw examples of that with like lactose being present in their environment or tryptophan being present. In eukaryotic cells, that also does occur where your guys' cells are sometimes responding to external stimuli, and then that causing a change in the expression of certain genes. But it's also important for a much bigger reason that's not true in prokaryotic cells. So in eukaryotic cells, now we're talking about multicellular organisms that are made of many, many, many cells. And the cells that make up a eukaryotic organism, they're differentiated and specialized. And so you guys are not just a clump of cells that are all the same. You guys are a clump of very specialized cells that are organized, forming different types of tissues and organs, for example. And those cells in a eukaryotic organism, they all have a copy of the entire genome. So all of your cells have a copy of all of the DNA. So all of the genes are in all of the cells. However, not all of the cells in your body are using all of the genes. And those different cells in your body are actually using different sets of genes. And so, for example, like you guys have nerve cells, and you guys have liver cells and skin cells and muscle cells, and cells that line the intestine of your stomach. You have all these different types of cells, but all of those cells are actually genetically the same. They have the exact same DNA. The DNA in any of those cells is exactly the same. However, the genes are being regulated differently in those cells. And so the genes that are being used by a nerve cell are going to be a completely different set of genes that compared to what are being used in like a liver cell. And so by genes being regulated in eukaryotic cells, this allows cells to become differentiated and specialized. And so now you can have a situation where everyone has the same DNA, but since you're all using the DNA differently, you can end up with different types of cells. And so that's a huge thing that needs to be accomplished in eukaryotic cells, making gene regulation super important. And so we're going to talk about ways in which genes are regulated and some of the different mechanisms that help regulate the genes in eukaryotes. You're going to see a lot of differences compared to what we saw in prokaryotes, and then maybe some slight similarities. But one thing we're going to start with is the fact that in eukaryotes, your guys' DNA exists as chromatin, which this is not true in prokaryotes. So in eukaryotes, you guys have, your DNA is actually wrapped around a bunch of different proteins. These are called histone proteins. And so in your guys' in the nucleus of the cell, for example, all of your guys' DNA is wrapped around these proteins to help condense the DNA and fit it inside the nucleus. We have a lot of DNA. And so your DNA is tangled up and wrapped around these different histone proteins. We call this chromatin. So chromatin is all of your DNA wrapped around these different proteins. They form these structures that look like beads on a string. Those are called nucleosomes. Nucleosomes are these histone protein structures that look like these little beads with DNA wrapped around them. And this is not something we see in prokaryotes. Prokaryotes don't have chromatin. They just have DNA, and that DNA is not wrapped around a bunch of proteins like we see in eukaryotes. And so the reason this is important is this actually on the chromatin level, actually there's some gene regulation that takes place. This helps control which genes are being used. Soon in this video, we're going to look at a lot of different levels at which gene regulation is taking place. But we're going to start on this level, which is the chromatin structure. The structure of your guys' chromatin. Your guys' chromatin, or the chromatin in eukaryotic cells, can be very tightly packed together. And some of your chromatin is a lot more loosely organized. And so there's two terms for that. Heterochromatin refers to regions of your guys' chromatin that's very tightly packed together. And then there's euchromatin. Euchromatin refers to these regions in your chromatin that are a lot more spaced out and more loosely organized. And this actually leads to certain genes being accessible and not accessible. So if there's genes in the heterochromatin regions of your DNA, those genes are going to not be accessible by like RNA polymerase. Like we're not going to be able to do transcription of those genes. Like we can't even access those genes because they're all tightly hidden in this packed structure here. But in the euchromatin areas of your chromatin, there we can access those genes in this region, and that's where transcription can take place. So already right off the bat, depending on which regions of your chromatin are tightly packed together as heterochromatin or loosely organized as euchromatin, is already going to determine which genes are even accessible for transcription to take place. And we actually see this in different types of cells. In different types of cells, like in a liver cell, there's going to be different regions that are packed together, which are going to contain genes that are not going to be expressed or transcribed by the cell. And there's other regions that are more loosely organized in your chromatin. But that's going to be different than in a heart cell or a skin cell or a nerve cell. So all of your cells might have the same DNA, but the way that DNA is structured in your chromatin, and which regions of that DNA are tightly packed or more loosely packed, is already going to cause different cells to be able to access different genes. And so this is kind of level one of gene regulation in eukaryotes, like which genes are even accessible, because if they exist as heterochromatin, you're not going to be able to access those genes, and if there's genes in there, you're not going to know about them in the cell. The cell's not going to be able to transcribe and translate those genes and express those genes. And actually in cells, they can actually change the structure of their chromatin. So they can cause certain regions of the chromatin to become more tightly condensed and other regions to become less tightly condensed. And this can change in a cell. So in a cell, in any given moment, there are mechanisms that allow them to take maybe heterochromatin regions and allow them to start loosening it so that they can access those genes. Or loose euchromatin regions, they can change it in a way where it becomes more tightly compacted and you can't access those genes. So the chromatin structure actually can change. It's can't think of words right now, but I'm really blanking on talking right now, but there's plasticity in your chromatin structure where it's not set in stone in certain regions, and other regions can become more condensed or less condensed in cells, which can make new genes accessible and other genes no longer accessible. And so here are a couple examples of how the chromatin can be modified to make the regions more or less compacted. So there's something called histone acetylation. Histone acetylation refers to when there's these chemical groups added to the histone tails. So these histone proteins have these tails that you see in this picture. These are part of the histone proteins that the DNA is wrapped around. And there's actually chemical reactions that can take place, or enzymes that can put these little acetyl groups on the histone proteins. So these histone proteins on their tails, there's these chemicals, these little chemical molecules called acetyl groups that get added to those histone tails. So we call that histone acetylation. We're adding these acetyl groups to the histone proteins. And in many situations, that actually causes the chromatin to become less tightly compacted. So it actually causes your chromatin to loosen up. So if the histones are acetylated, that can allow some of this heterochromatin to loosen up as euchromatin. And now we have areas of DNA that are now accessible for transcription. An opposite example of that is DNA methylation. And so another thing that could happen is there's enzymes that can take pieces of your DNA and add these methyl groups to it. Specifically, usually it's the cytosine bases in your DNA. There's enzymes that can add these chemical groups called methyl groups to them. And we call that methylation. And so these little red dots represent these methyl groups that are being put on your DNA. And when those methyl groups get added to your DNA, in a lot of situations, it causes the chromatin to become more tightly compacted. So when the DNA is methylated, the chromatin tightens up and becomes a lot less accessible for transcription. Whereas when those methyl groups are not present, it's a lot looser and exists more like euchromatin and less like heterochromatin. And so that's the just two examples of how the chromatin structure can change depending on if we're adding these acetyl groups to the histone proteins or these methyl groups to the DNA, making certain regions more condensed or less condensed. And this is going to affect which genes are accessible for transcription, which genes we can actually use to make mRNA, which then we can then send to a ribosome to make proteins. And so these changes that are made to your guys' chromatin structure are called epigenetic changes, which are going to alter the expression of your guys' of phenotypes in an organism without technically changing the DNA. So we're not mutating the DNA or changing the DNA, but we are changing the structure of the chromatin, either making it more compacted or less compacted. And by doing that alone, we can already change which genes are being expressed in a cell. So those are called epigenetic changes without changing the actual DNA. And so beyond that, so think of this as like level one, like the chromatin structure in a eukaryotic cell. Which regions are less condensed, which regions are more condensed. And so you're already going to have now in level one certain regions of DNA that are accessible and certain regions of DNA that are not accessible. Then we could talk about level two, if we want to call it that. And level two of gene regulation in eukaryotes. Now, let's say there is genes located in the euchromatin area of your chromatin. So this is a gene located in the less condensed region of your chromatin. So let's say it is accessible, like it exists in like these areas that are less condensed. Well, now there could be these regulatory sequences that help now determine whether or not that gene will actually be expressed or not. And so regulatory sequences are special sequences in your DNA that proteins can bind to, and those proteins binding to those regulatory sequences will affect and help determine if genes get transcribed or not transcribed. And so we've seen some examples of regulatory sequences already in prokaryotes, and some of them apply also to eukaryotes. Like promoters are sequences in your guys' DNA. It's actually like pieces of your DNA that RNA polymerase binds to. We call that a regulatory sequence. RNA polymerase is a protein that can bind to that sequence and then obviously cause transcription to start happening. In bacteria cells, in the last set of notes, in topic 3, we talked about operators. Operators are also regulatory sequences. These are sequences in the DNA of prokaryotes that repressor proteins bind to when they're active. And so these regulatory sequences become important in helping determine which genes are going to be transcribed or not. And in eukaryotic cells, we'll talk about these in a second. There's things called enhancer sequences that activator proteins can bind to, which will help stimulate the transcription of certain genes. And so here's a kind of like a picture showing you a typical eukaryotic gene. So here's a gene here. Let's say this is the gene, and you guys can see there's introns in this gene that are these non-coding regions. But let's say this is a certain gene with the instructions to build a protein. There's the promoter sequence, where RNA polymerase will bind to. So that's an example of a regulatory sequence. And then there's these other regulatory sequences. Some of them are close by, some of them are far away in your DNA, not near this gene. But these other sequences in your DNA, which are non-coding, they're not actually genes, but these are areas that proteins can bind to. And when proteins bind to these regulatory sequences, it's going to affect, or it can affect, whether or not there's transcription of this gene over here. And so that's what we're going to talk about right now as kind of some examples of that. So again, here's a promoter sequence. A promoter sequence, this is present in both prokaryotes and eukaryotes. These are sequences in the DNA that RNA polymerase will bind to, where indicating where a gene is, so that RNA polymerase knows where to go start doing transcription and where to start making the mRNA. In eukaryotes, I mentioned this before, but just to point it out again, in eukaryotes, our promoter sequences often have these TATA boxes built in, which is just an area that has a bunch of adenine and thymine base pairs. And so what happens is RNA polymerase will bind to these promoters, but there's also these other proteins that get involved with the promoter sequence called transcription factors. And we didn't really see this in prokaryotic cells. In prokaryotic cells, we didn't really talk about transcription factors, but these are proteins that are going to bind to, in this case, the promoter sequence, and are going to help RNA polymerase bind to the promoter sequence. So in eukaryotes, it's a little bit more complicated. RNA polymerase won't just go and bind to a promoter all by himself. He needs the help of these transcription factors to find the promoter and bind to it successfully to start initiating transcription. So these transcription factors become very important in eukaryotes to help start the process of transcription and ultimately expressing certain genes. And so there's two types of transcription factors that exist. Well, we could classify transcription factors into two categories. There's things called general transcription factors and specific transcription factors. General transcription factors, these are proteins needed to do transcription of any gene. So for any given gene, there is a set of transcription factors you must have binding to the DNA in order to get that gene expressed, in order for transcription to happen of that gene. And that would be true for any gene, for all genes. You would need this special set of transcription factors. We call that general transcription factors. They're needed in general for all of your genes. Because there is a group, like I was mentioning earlier, like RNA polymerase, there is a group of proteins, transcription factors, that have to be present in order for RNA polymerase to even bind to this DNA and start transcription. But then there's also transcription factors called specific transcription factors that are going to be specific for certain genes. So certain genes, on top of needing general transcription factors, which all genes need, there's other transcription factors that can come into play just for specific genes. And so for certain genes, there's general transcription factors that are needed, but sometimes there's these other specific transcription factor proteins that will get involved for that specific gene. And those specific transcription factors can either act as activators or repressors, which is very similar to what we saw in prokaryotes. There's activator proteins and repressor proteins that can bind to the DNA and either increase or decrease expression or transcription of certain genes. And so the activator proteins, they will bind to regulatory sequences called enhancers. So enhancers are sequences in your DNA that activator proteins bind to. And then there's regions called silencers, or regulatory sequences called silencers, that repressor proteins bind to. So enhancers and silencers, these are sequences in your DNA that repressors and activators bind to. So here's a picture kind of illustrating some of this. So here we have a gene right here, the beginning of a gene, and this is the promoter sequence. And so you can see here is RNA polymerase binding to the promoter sequence. But on top of that, there's look at all these other proteins in this picture. It gets really messy very quick. And so a lot of these proteins are general transcription factors. Like a lot of these are transcription factor proteins that must be present for any gene to be transcribed. For RNA polymerase to come to this promoter and bind to the promoter and start transcribing the gene, there is a collection of transcription factors you're always going to need. We call those general transcription factors. And this is this is very specific for eukaryotes. In prokaryotes, we didn't see all these transcription factors that were needed. But in eukaryotes, we do need a bunch of these transcription factors. Now, on top of those general transcription factors, there's also specific transcription factors that get involved only for certain genes. So for this specific gene, there's some activator proteins that are binding to these enhancer sequences. So enhancer sequences are sequences in your DNA. These are regulatory sequences that activator proteins bind to that will affect the expression of this gene. And then right here is a silencer sequence. That's a sequence in DNA that repressor proteins bind to. So these terms that I'm using these are terms that we very specifically usually use for eukaryotic DNA. So when I say enhancers and silencers, I'm talking about sequences in your DNA that activator proteins are binding to and repressor proteins are binding to, which are specific transcription factors. Even the word transcription factor, that's usually a term used usually for eukaryotic genes, okay? These proteins involved in expressing eukaryotic genes. And so a repressor protein, if it binds to the silencer, that will help to hinder or stop transcription of this gene. But these activator proteins help to boost or encourage transcription of this gene. And so there's a lot of moving players here, a lot of different players involved in whether or not this gene will be expressed. So you're going to need all these general transcription factors to work with RNA polymerase, but sometimes you also need these activator proteins to get involved to help a gene be expressed. And then on top of that, there's sometimes repressor proteins that can get involved for certain genes as well. And so try to start thinking about all these vocab terms correctly. So enhancers and silencers, which are sequences in your DNA that repressors bind to or activator proteins bind to. And then you have these general transcription factors, which are always needed for any gene, for every single gene, for RNA polymerase to start transcription. And this becomes, well, first of all, complicated. We're going to try to simplify it for the purpose of this class, though. I'm going to focus on these activators. So a lot of times, whether or not a gene gets expressed has to do with if these activator proteins, these specific transcription factors, are present and binding to these enhancer sequences. Because a lot of times, if you don't have those activator proteins, so here's a picture over here showing like, okay, here's a gene and a promoter. And then here's the enhancer sequences that specific activator proteins have to bind to. And when those activator proteins bind to these enhancer sequences, and when the general transcription factors come and bind to the promoter and help RNA polymerase get started here, all together they will work together to start transcription of a eukaryotic gene. But this will only happen if these activator proteins become really important in eukaryotes. If these activator proteins are not here, usually that will cause the gene to not be expressed. Like you have to have these activator proteins, these specific transcription factors present in order for this gene to turn on. And if these guys weren't here, then what likely would happen is none of this would happen. Like we wouldn't be able to start transcription of this specific gene. Okay, so for the purpose of this class, at least, we're going to focus a lot on activator proteins and whether or not they're there and which activator proteins are needed for this specific gene. And if they're there, you're good to go. If they're not there, well, then that gene's not going to turn on. Here's an example, and this one's in one of the questions in your guys' notes. I even recently have seen this in an AP question, the same exact situation. So I kind of want to walk you through what's going on in this picture here. So this picture is comparing a liver cell with a lens cell. And so liver cells are cells that make up your liver. And then your lens cells, those are cells found in the lens of in the front of your eye. So these are two cells you have in your body, very different types of cells found in your body. And these cells do very different things inside of your body. And then what they're looking at here is two different genes present in the DNA. They're looking at this gene for albumin. Albumin is a protein that's made in the liver cells. And then they're looking at this gene called the crystalline gene. Crystalline is a protein that's made in lens cells. It's a transparent protein that allows light to go through. Albumin, on the other hand, is actually a really opaque protein. It's very foggy. Like if you had a solution of albumin, it would be very gross and foggy. And so this is not a protein you would want to make in your eye, like in your lens cell. That would block your vision a lot. But here's the thing. Okay, let me go over some important key concepts here. First of all, liver cells and lens cells have every single gene in the human genome. So in your liver cell is all of your DNA. So if I had one cell from your liver, I would see all of the DNA that exists with every single gene. Every single gene is in the liver cell. And in the lens cell, every single gene is in the lens cell. So both liver cells and lens cells have these two genes. Like in the liver cell, you would have both these genes because you have every gene in the liver cell. And in a lens cell, you have both these genes because you have every gene in a lens cell. However, in liver cells, the albumin gene is turned on and being used, and the crystalline gene is not turned on and not being used. Even though both those genes are present, you're actually only using the albumin gene, you're not using the crystalline gene. And in lens cells, it's the opposite. The crystalline gene is turned on, and we're making the crystalline protein, but we're not making the albumin protein. So the question becomes, why is that happening? Why in liver cells does it make this gene and not this gene? And why in lens cells are they making this gene and not that gene? A common incorrect answer that students give sometimes, please never say this on an AP test, is that Well, the reason why Lin cells make the crystalline protein and the liver cells make the albumin protein is because liver cells have the albumin gene and Lin cells have the crystalline gene. That's a terrible answer because all cells have every gene. The liver cells have both the albumin and the crystalline gene. The Lin cell also has the albumin and crystalline gene. So that doesn't explain why one of those proteins is being made in one of the cells and not the other. It has to do with which of these genes is being expressed. And in liver cells, only the albumin gene is expressed, and in Lin cells, the crystalline gene is expressed, and not vice versa. And so here's what's actually going on. In a liver cell, so let's say we look at a liver cell. In a liver cell, there's going to be certain activator proteins floating around. These are specific transcription factors. There's certain transcription factors that are present in a liver cell, okay? And specifically, some of them are the ones needed for this albumin gene. Here are the enhancer sequences for the albumin gene. And in order for this gene to turn on, you have to have the activator proteins bind to these enhancer sequences. And each of these enhancer sequences requires a different version of a different activator protein, a different specific transcription factor. So they kind of color-coded in this picture. You can see like this guy can bind to that enhancer, and this gray guy can bind to that enhancer, and this yellow activator can bind to this yellow enhancer. And so if you have those three activator proteins present, they can each bind to their enhancer sequence, which will allow the albumin gene to turn on. So in liver cells, they must have, if the albumin gene is on, that means the transcription factors needed for the enhancer sequences for the albumin gene, those transcription factors must be present in a liver cell. That's the only way this gene would turn on. But if you look at the crystalline gene, there's a different set of enhancer sequences. Okay, it's different than these enhancer sequences. You have this enhancer sequence, this one, and this one, which require a different set of specific transcription factors, a different set of activators. And you can see in a liver cell, you don't have any of these guys. You don't have this guy or this guy or this guy that are able to bind to these enhancer sequences here. And so that crystalline gene will not be expressed. And so the reason the albumin gene is being expressed and the crystalline gene is not being expressed, a lot of times it can be explained by which activator proteins, which specific transcription factors are present in a liver cell. And if you have some of the ones needed for the albumin gene, then that gene can turn on. But if you don't have all the ones needed for the crystalline gene, that gene's not going to turn on. And if you look at a Lin cell, the big difference here is in a Lin cell, they have a different set of activator proteins floating around. In a Lin cell, the activator proteins that are available look a little bit different. There's a different collection than what was present in a liver cell. In a Lin cell, you have some of these guys and those guys and these guys, which are not all the ones you need to turn on the albumin gene. You're missing this yellow one, the one that goes to the activator that binds to this yellow enhancer, and you're missing the one that binds to this enhancer sequence. Like they're not floating around anywhere in a Lin cell. In a Lin cell, they don't have those specific transcription factors. So this gene is not turning on. But you do have the ones you need to turn on the crystalline gene. So you do have this pink guy floating around and this gray one and this brown one. These activator proteins that can bind to these enhancer sequences causing this gene to turn on. And so the correct explanation for why in a liver cell, it makes the albumin gene, but not the crystalline gene. And in a Lin cell, it makes the crystalline gene, but not the albumin gene, has to do with which transcription factors are present in a liver cell and how that's going to be a different set of transcription factors that are different than what's present in a Lin cell. And depending on which transcription factors are available, you can, you're only going to have certain ones that can bind to certain enhancer sequence present. And if you don't have all of the ones needed for certain enhancer sequences, certain genes won't be able to turn on. You can only turn on the gene if you have all of the activator proteins needed to bind to those enhancers for that gene. So I hope that, I don't know, it's not maybe the most concise way I could explain it, but hopefully this picture makes a lot more sense now and why certain genes are turning on or off depending on which specific transcription factors are available in that cell. So that's another level here where gene regulation is taking place. So even if a gene is like in the euchromatin region of your DNA, like let's say you have genes here and they are accessible. Well, first of all, yes, they have to be accessible. But the next question becomes, do you have all the transcription factors you need to turn on that gene? Because if not, that gene will still not be on. So not only does the gene have to be accessible in the euchromatin areas of your chromatin, it also has to have, in the cell, there has to be the transcription factors needed for that gene if you want that gene to turn on. And so the big takeaway here is that activator proteins become really important in eukaryotes. And if they're not all present for a certain gene, that gene will not turn on. And different cells have different collections of activator proteins. So anyway, and then there's just a couple more things in here to kind of dissect together. Another thing it talks about in here is how genes are coordinated, how the expression of certain genes is coordinated in eukaryotes, and how this is going to be different in prokaryotes. So coordination of gene expression refers to the fact that sometimes you have certain genes that work together to accomplish something important inside of a cell. And so we saw this with like the bacteria example, the prokaryotic gene regulation when we looked at operons, like the lac operon and the trip operon. And the trip operon, there's 5 genes, and those genes create proteins that all work together to make tryptophan. And so it becomes really important that if you're making one of those genes, you make all of those genes because they all work together. Well, in eukaryotes, there's very often you guys have genes that work together, the genes that make proteins that work together to do something inside of your cells. And it becomes just as important that those genes are being expressed together, because you don't want to make gene A and gene B and then not make gene C, when to do this one thing inside the cell, you need all 3. You need gene A, B, and C. You need all 3 of those proteins to do this one task inside the cell. You're going to need all 3 of those genes. And so the question becomes, how in eukaryotes do they make sure that genes that work together are expressed together? And it's a lot different than what happened in prokaryotes. In prokaryotes, the answer was kind of easy. In prokaryotes, a lot of times they are grouped together sharing a promoter. So because they all share a promoter, when any of those genes are expressed, they all will be expressed. Like we saw in the trip operon and the lac operon, like those groups of genes sharing a promoter allows them to always be expressed together. Super convenient. Now in eukaryotes, here's the big difference. In eukaryotes, you guys would never have genes grouped together sharing a promoter. That does not happen in eukaryotes. Never in eukaryotic DNA do multiple genes share the same promoter. In eukaryotic DNA, every gene has its own promoter. So every gene is actually independent of each other. They're never going to be grouped together sharing a promoter. So this becomes trickier. So what happens, or one main way that coordination takes place, it has to do with these transcription factors again. And usually what you find, if there's 3 genes that work together, like let's say this gene 1, 2, and 3, produce these 3 proteins, and these 3 proteins together work together to accomplish something important in the cell. And you need all 3. Like you would never just need protein 1 or 2 without 3, or 2 or 3 without 1, or just 1 by itself. Like you need all 3. And so the way a lot of times these genes get coordinated together is the transcription factors that are needed, the specific transcription factors that are needed for gene 1. Let's say gene 1 needs transcription factor A, B, C, and D. And maybe these are specific activator transcription factors, activator proteins, that are needed to turn on gene 1. Well, what we find usually is that genes that work together with that gene also require the same set of transcription factors. So gene 2 also requires A, B, C, and D to turn on gene 2. And gene 3 also requires those transcription factors. And so now we have a situation where if one of these genes turns on, so if you're in a cell and you have transcription factor A, B, and C present, well, that's not enough to turn on gene 1, but it's also not enough to turn on gene 2 or gene 3. Like if these are activator proteins, let's pretend these are activator proteins. If you need all 4 of these activator proteins to turn on gene 1, and one of them is missing, this gene won't turn on. But this gene also has the same set of requirements, so this gene won't turn on, and this gene won't turn on. Like those 4 transcription factors, activators, are needed to turn on all of these genes. But if you're in a cell, and in that cell you do have activator A, B, C, and D floating around, there's a bunch of these guys floating around, and these guys floating around, and these guys floating around, and these guys floating around, that will successfully now cause gene 1 to turn on. Those are all the activators you need to turn on gene 1. And since gene 2 and 3 have the same requirement, those genes turn on. And so by requiring the same set of transcription factors, genes can be expressed together or not expressed at all. Like if gene 1, 2, and 3 all require the same set of transcription factors, well, that means if you're in a cell and the transcription factors that are needed to turn on one of those genes is present, well, then the other genes would also be on because they require the same set of transcription factors. And if you're in a cell where not all of these guys are present, well, then none of these genes would be on because, again, they all require the same set of transcription factors. Think of these specific transcription factors, like these activators, as like the keys needed to unlock certain genes. And for this gene, you need 4 different keys, all of these guys, to unlock gene 1 to turn gene 1 on. But that's the same set of keys you need for gene 2 and gene 3. So this will create a situation where if one of these genes is able to turn on in a cell, well, then the other ones should also be able to turn on because they have the same requirement for the keys or the transcription factors. And so this is how genes get expressed together in eukaryotic cells. A lot of times they have the same set of activator proteins that they need to turn on those genes, which a lot of times is in the form of these enhancers. Like the enhancers for gene 1, 2, and 3 would be the same for each of those genes, requiring the same activators. And so if those activators are there, those genes will all turn on. If those activators are missing, none of those genes will turn on. So this is, hopefully that kind of makes sense, but that's how genes are being coordinated. The expression of genes are being coordinated sometimes in eukaryotes, how we can make sure that certain genes that need to be expressed together are always expressed together or not expressed at all. And by requiring the same set of transcription factors, that kind of creates a situation where, yeah, if one of these genes is able to turn on, they all should be able to turn on. Or if one of them is not able to turn on, well, then none of them are on. So either they're all being made or none of them are being made. And so the last thing on here is the fact that in eukaryotes, there's also things that you can do after transcription to control which genes are being expressed. And that's called post-transcriptional regulation, so after transcription. So let's say there was a gene, and let's go through all these levels now. Like let's say that gene was in the euchromatin area of your chromatin, so meaning that it was accessible. And let's say you had all the transcription factors needed to turn on that gene. And so now RNA polymerase is making mRNA and transcribing that gene. And now you have the mRNA for that gene. Well, even after you make the mRNA, there's still ways you can manipulate or affect whether or not that gene gets expressed, because you haven't fully expressed the gene yet. You've transcribed it to mRNA, but you still have to send that mRNA to a ribosome to then translate it. And so there are things that can happen after transcription to still prevent a protein from being made, even if you did transcription. And so there's 2 examples I want to show you. One of them is alternative RNA splicing, and the other one's called RNA interference. And these are the last 2 things in the notes. And so for alternative RNA splicing, if you guys remember, in eukaryotic genes, in a eukaryotic gene, there's going to be these exons and introns embedded in the gene. And these intron regions, which are not labeled here, these are the non-coding parts of the gene. And these exons actually have the instructions to produce the protein. So let's say this is a single gene here, and you can see it's kind of split up by these different introns. And so when you make the mRNA, you copy all of it, all the introns and exons. And what you have to do, if you guys remember in eukaryotes, is you have to splice out the introns, these non-coding pieces, and put together the exons. And so what happens, though, is sometimes there's certain genes where you can splice out the introns along with some of the exons and end up with different combinations of exons being spliced together that can actually lead to different proteins being made when that mRNA gets sent to the ribosome. And so if you guys look here, what's happening, like in this situation here, they spliced out all the introns, but they also spliced out exon number 3. And so they put together exon 1, 2, 4, 5, and 6. And if you send this mRNA to a ribosome, it's going to read that mRNA and all the codons and put together the amino acids, and you end up building this certain protein here that has a certain function. But that mRNA sometimes can be spliced in different ways. Like in this case, there's also a way you can take this mRNA and splice out the introns and also splice out exon number 2 and 4 and put together exon 1, 3, 5, and 6. And then you end up with this mRNA. You send that to a ribosome and it's going to result in a different protein being made. Like the codons here are now going to be lead to a different protein than all of this right here. And so that's what this is showing over here too. Like you could splice out the introns along with exon number 2. So alternative RNA splicing is when the RNA gets spliced differently sometimes into different combinations of exons. And those exons, when they're translated, lead to different final proteins. And these final proteins might have slightly different functions inside of the cell. So here's a situation where we had a single gene, like this was just one gene, but we ended up making like 3 different proteins from that gene, just depending on how we spliced the mRNA. And this happens sometimes in eukaryotic genes. There's some genes that exist, scientists have found, certain genes that actually code for more than one protein. And that's possible because of how the RNA can be spliced differently sometimes, which actually solved a huge problem, because what they were realizing when they started studying DNA in different organisms, like in humans, for example, is they started realizing that the amount of proteins being made, let's just say in humans, the amount of proteins that are made inside of human cells outnumbers the amount of genes that are in our DNA. So in our DNA, there's like 20,000. It's estimated that there's about 20,000 genes in our DNA. In the 46 chromosomes that you have, there's 20,000 genes. But what they were finding is that there's actually a lot more than 20,000 proteins. If we added up all the different proteins that are made in all the different types of cells that you have, there's more than that. And for a long time, it was kind of a puzzle. Like, how is that possible? How can you have more proteins being made than genes? Because genes code for proteins. So how could you end up with more proteins than there are genes? And what they discovered at one point is that this thing called alternative RNA splicing exists, which now creates a situation where a single gene can actually lead to multiple different proteins depending on how it's spliced. Okay, so this is kind of a cool ability where we can actually make 3 different proteins all from the same gene by splicing together the exons in different combinations. And then the last thing here talks about RNA interference. And this is a situation where small pieces of RNA can bind to mRNA and block the mRNA from being translated by a ribosome. So sometimes you do successfully make messenger RNA, and you're really excited that messenger RNA was transcribed, and now it's going to be sent to a ribosome to be translated. But sometimes what happens is these small pieces of RNA called microRNA, they can go find certain pieces of certain mRNAs that they can bind to complementary. And by binding to them, they actually cause that mRNA to not be translated by a ribosome. It blocks the ribosome from reading this mRNA, and then usually leads to that mRNA being destroyed. And so then that mRNA gets broken down and degraded, and then we technically never made it past the finish line. We never expressed this gene, the instructions on this mRNA. We didn't finish it. I mean, we transcribed it, but we never translated it, so we never made the protein. And so sometimes these little microRNA things get in the way and can stop a gene from being expressed as a protein even after transcription takes place. So just because you finish transcription and make the messenger RNA, you're not safe yet. Like that gene, we can still stop that protein from being made, such as through RNA interference, where these microRNA pieces, these small pieces of RNA, can sometimes find certain pieces of mRNA that they can bind to and prevent them from being translated. So another way that we can ultimately control what genes are being expressed in eukaryotes. So to summarize, try to give one final summary. If there's a gene, if the question is, okay, is this gene in a eukaryote, is this gene going to be expressed? The answer to that question is very complicated. Like, is this specific gene in this cell going to be expressed? Well, things that would have to, in order for a gene to be expressed in eukaryotes, first of all, starting on the chromatin structure, that gene has to exist in an area of the chromatin that's not tightly compacted. It would have to, not in a heterochromatin region, but in a euchromatin region. And just to kind of recap some of these pictures here, like, first of all, a gene would have to exist here, not here. Now, if a gene does exist here, you made it past level 1, but then you also have to have these specific transcription factors present to bind to these enhancer sequences for that gene to actually be transcribed by by RNA polymerase to make the mRNA. And if you don't have those activator proteins present, well then it doesn't matter if the gene is accessible. Even if the gene's accessible, if you don't have these activator proteins, then the gene won't be expressed. But let's say the gene is accessible in your chromatin and you have the activator proteins needed to transcribe the gene, and now RNA polymerase comes and transcribes the gene into mRNA. Well, then that mRNA needs to now not be blocked before it gets to a ribosome, and then before the ribosome has a chance to actually make that protein. So there's a lot of different levels that are controlling whether or not certain genes are being expressed in eukaryotes. And there's actually, we could go into way more details. There's a lot of different mechanisms, but here are just some different examples of mechanisms at different levels controlling whether or not in eukaryotes a gene is being expressed or not being expressed. Okay, so it's a lot more complicated, unfortunately, than prokaryotes, but hopefully you now know some of these examples about the chromatin structure and these transcription factors being present or not being present, and things like RNA interference that can still prevent a gene even after transcription from being expressed. And so that's the kind of like a more general overview, even though it sounds like a lot, of gene regulation in eukaryotes, because there's a lot more things we could talk about on in terms of how a gene is regulated in a eukaryotic organism. But that's the overall gist of some of these different components controlling gene regulation. I hope that makes a little bit of sense. We'll talk about this a lot in class, obviously, and kind of continue dissecting some of this information. But just kind of carefully go through that, try out some of these questions in your notes, and we'll talk about it soon. Anyway, thank you guys. I'll see you later.